Bismillahirrahmanirrahim. Today I would like to share the knowledge about PCR primer design guidelines, and this is the important for the researcher, because when the researchers are doing the PCR, then the first step is that they should know about the PCR primers, and it is the most important step for the PCR technique. So it is important for the researcher to get the knowledge about the PCR primer designing. How can the researcher make the PCR primers? So this one is what is the PCR? It is the polymerase chain reaction, one of the most important inventions of the 20th century in molecular biology, and a small amount of the genetic material can now be amplified to be held to following reasons: and identify, manipulate the DNA, detect the infectious organisms, include the viruses that cause the AIDS, hepatitis, tuberculosis, detect the genetic variations including mutations in human genes, and numerous other tasks. Here is the little sketch. Double stranded, that the DNA, which is when you perform the PCR technique, then the three main steps are involved, like the denaturation, annealing, extension stage, and at the annealing stage the primers are important, because these primers actually play important role to extend the to multiply the DNA strands. So this is the actually introduction of the PCR, but here is the PCR primers. How can you define it? It is the single strands of nucleic acid, synthetic nucleotide, oligonucleotide that is necessary to initiate the PCR. So these are the specific and complementary to the target DNA or RNA sequence portion. Here is the little see here that these are the primers which are attached to these 5' to 3, and here 3' to 5'. So these are the primers, these are the short fragments. So primer design, it is essential for successful reactions. The important design considerations described below are a key to specific amplification with high yield. So the preferred values indicated are built into all our products by the default. So this one is the first step, primer length. It is generally accepted that our optimal length of the PCR primer is the 18 to 24 base pair. This length is long enough for adequate specificity and short enough for the primers to bind easily to a template at the annealing temperature. So here is the little sketch of these primers. Next one is the primer melting temperature. It is also the most important. The primer melting temperature, by the definition, is the temperature at which half of the DNA duplex will dissociate to become a single strand, and indicates the duplex stability. Primers with melting temperature in the range of 52 to 58 degrees centigrade generally produce the best results. The primers with a melting temperature above the 65 have a tendency for the secondary annealing step. So the GC content of the sequence gives a fair indication of the primer TM, and all our products calculate it using the nearest that the neighbor thermodynamic theory accepted as such as a much superior method for estimating it, which is considered the most recent and the best available. So the formula for primer design TM calculation: the melting temperature TM into Kelvin, that delta H over delta S plus R in incorporation to the C. And next is our situation: the melting temperature Tm is equal to delta H over delta S plus R into C, that minus 273 plus 15, where the delta H is the Kelvin per mole, H is the enthalpy and amount of the energy. Delta H is in a change the enthalpy in above the formula of delta H strained. So this formula you can apply this, and next is you can see melting temperature. The primer that melting temperature is estimate of the DNA hybrid stability and critical in determining the annealing temperature. Too high and TA will produce the insufficient primer template hybridization, results in the low PCR product yield. Too low and TAM may possibly lead to that the non-specific product caused by a high number of the base pair mismatches. Tolerance is found to have the strongest influence on the PCR specificity. So this is the formula. Next is the GC content should be 40 to 80%, and XC clamp, the presence of the G or C base within the last 5 bases from 3 end of the primer GC clamp that helps to promote the specific binding at the 3 end of the stronger bonding of the G in and C bases. More than 3G and C should be avoided. This one you can see here, this the 5 prime to 3 prime, that the G. Then next is the primary secondary structure, and you can see here that the next is that is for molecular interaction with primer and the self-dimer. Primer self-dimer is formed by the intermolecular interaction between the two or same sense primer, where the primer is homologous to itself. So next is the cross dimer are formed by the intermolecular interaction between the sense and anti-sense primer, where they are homologous. You can see in this structure. And next is the repeats. Dinucleotide occurring many times consequently and should be avoided because they can misprime. So runs with long runs of the single base should generally be avoided as they can misprime. So this is the step of that. Then the end stability, you can see here it is the maximum, that the delta G value, and avoid the template secondary structure. So next avoid the cross homology, and to improve the specificity of the primer it is necessary to avoid the region of the homology and specific other mistakes. So the structure you can see here how to binders. So this is the camely that the homology. The specificity of the primers is necessary necessary to avoid the regions of the homology. So these are the steps. Commonly, these primers are designed and then blasted to test specifically our product that offer a better alternative.